Okay, hi everyone. I am Ophir Geminder. I'm an MD PhD student from the Hebrew University in the lab of Professor Mayam Salton. And I want to show you today some of the work that we do in lab about the WBB4 spliceosome subunit and its role in neurodevelopment. So our story starts with a family of two first cousin parents with a child that has this syndromic autism spectrum disorder. And a trio exome test of the family discovered that the child is homozygous for a mutation in the gene named WBB4, while the parents are heterozygous for the mutation. And the mutation that the child has is a deletion of one nucleotide in the WBB4 gene, which causes a shift in the reading frame and causes the mutation in the gene. Following up the family, they had multiple miscarriages. And the most recent one was after we started the research, so we sequenced also the fetus, and the fetus also had the mutation in the WBB4 gene as well, the homozygous mutation. Using GeneMatcher, which is the tool that allows us to upload gene that we are studying and contact with other groups around the world to work together and share data, we found a total of 12 probands with homozygous mutations in the same gene, in WBB4. And we recently published the paper about all these probands, and all of them, and they also had facial dysmorphic features and other phenotypes as well, such as intellectual disability, motor delay, speech impairment in 10 out of 10 that we could test these features. And out of 5 that were assessed for autism spectrum disorder, 4 were indeed diagnosed. Using fibroblasts from 2 of the families, from the proband of the family and the 2 parents, we could see that the WBB4 mutations cause loss of the WBB4 protein expression, as we could validate in Western blot from these fibroblasts. And after talking so much about WBB4, I want to tell you what it does. So WBB4 is a spliceosome subunit. Spliceosome is the complex that is responsible for the splicing process, which is the process in which we take the premature mRNA and we edit it into the mRNA by taking out the introns and conjugate together the exons. And this mRNA will later be translated into the protein. So WBB4 is part of the spliceosome that creates the mRNA from the pre-mRNA. And we usually divide the spliceosome activity into the different parts of the action. And WBB4 is part of the B complex of the spliceosome. What it does is it inhibits the BRR2 helicase, which is the helicase that does the unwinding of U4 and U6 SNRNPs. And this is what is required for the next step of the spliceosome. So WBB4 is stopping the next step of splicing until the right splice site is selected. So it kind of regulates when the splicing will take action after choosing the right place for it to happen. So we speculate that when WBB4 is missing, BRR2 helicase might act prematurely, and this will promote incorrect splice site selection and therefore impaired splicing. We also know from published data that WBB4 is highly expressed during neurodevelopment and specifically in early neurodevelopment, around week 10 of the post-conceptional weeks, which is a critical time for neurodevelopment. So we think that these splicing aberrations cause ASD. And we wanted to ask what could be the mechanism of these splicing aberrations to cause ASD. And to answer this question, we generated a mice model of WBB4 knockout in which we tried to mimic the mutation that we had in few of the probands. So what I told you in the first slides about the family that we started the whole story with that had a deletion of an nucleotide. So using CRISPR, we could create an insertion of one nucleotide in the mouse WBP4 gene, and this as well caused the frame shift of the reading frame. And as well, the WBP4 knockout mice mutation causes protein ablation, as we could see in this Western blot. But following the mice colony, we could see that we cannot detect any mutant mouse at the post-weaning phase. So we went back and we tried to look where we can see the mutants. And we could see some mutants during the pregnancy at different time points during the pregnancy, very few at P0, the day that the mice are born, but none at weaning phase at P21. So we could conclude that WBP4 knocked out mice do not reach adulthood, and they have embryonic and perinatal lethality. So after understanding that we will not have any WBP4 knocked out grown mice, we went back to day E15.5 of the pregnancy when crossing two heterozygous mice, and we looked at the embryos. So we could see that WBP4 knockout embryos are significantly smaller than both wild-type and heterozygous embryos, as you can see in the picture. And taking out the brains of the embryos as well, we could see that the brains are smaller as well. So WBP4 knocked out mice exhibit phenotypical changes, similar to what we saw in humans, that also has some phenotypical features. So because we couldn't have samples of WBP4 brains from humans, obviously, and we wanted to have some tools, some models that we can use to test and to see what and to study the mechanism, we initiated this mice model. So we took both models we have, the fibroblasts from the families and the frontal cortex of the embryos at day 15.5 of WBP4 knockout mice, and we deeply sequenced the RNA of them to study the splicing changes. And we wanted to see if the models have similar misspliced target genes. So doing RNA sequencing analysis for both models, human and mice, we could see that they have significant overlap of genes that are abnormally spliced. We have 814 genes that are the same genes having mis-splicing both in human and mice WBP4 ablation. And looking at the phenotypes and at the gene ontology enrichment of both models, we could see that for human, the top hits are abnormality of the nervous system, abnormal nervous system physiology, meaning relevant phenotypes to what we saw in human. And for mouse, we have complete embryonic lethality during organogenesis, decreased embryo size, decreased body size, and premature death, all aligned with what we actually saw. So WBP4 knockout mice and human splicing abnormalities target similar genes, which are relevant for the matching phenotypes. So what we were interested in is to see what are these genes, what are these target genes, and why they are so impacted from WBP4 absence. And later we took these 814 genes that are abnormally spliced in both models we have, and we crossed it with SFARI genes dataset, scores 1 and 2, and people before me had described this dataset, so I will not expand about this. And we could see that there is also an overlap of 71 genes, which is a significant result. So we can say that WBP4 splicing targets abnormalities in both human and mice happen also in ASD-associated genes. So we were really interested in these 71 genes, and we are still interested in it, to see what are these genes, why they are affected, and we want to use both the models we have to study this. And this is some validation that we did in the lab in order to see some very interesting genes that are also ASD-associated and change in both models, human and mice. that changed their splicing in the absence of WBP4. So I will summarize everything I just said. So when WBP4, so we hypothesize and speculate that when WBP4 is present, we have proper splicing of the relevant target genes that are relevant for neurodevelopment and ASD development. And then the pre-mRNA is turning into mRNA in the correct way. And then we have normal neurodevelopment and normal development and viability in mice. While when WBP4 is absent, we think that the pre-mRNA is incorrectly spliced. Then we have splicing alterations, which causes the neurodevelopmental delay in human and ASD as well, and embryonic lethality in mice. Thank you very much for the opportunity to show you, and I would like to take questions. And I know that the poster session is already over, but if you still want to see more data about this project and similar projects, so I have my colleague's poster out, which is 2 and 46, and you are invited to see it. And that's it. Thank you.